Hello everyone, welcome to the Medical Chaps. This video is about heme degradation and jaundice. So heme is released when RBC is burst in liver and spleen. In liver, this heme is ingested by the macrophages. Inside macrophages, there is an enzyme called heme oxygenase, which converts the heme into biliverdin. Iron and carbon monoxide are removed, whereas oxygen is added and NADPH is oxidized. Biliverdin is green in color. This biliverdin is immediately reduced into bilirubin through biliverdin reductase enzyme present in the macrophages. Bilirubin is now orange-yellow in color. This bilirubin is released by macrophages into the blood capillaries. Bilirubin then combines with albumin, as we know that bilirubin is hydrophobic. Therefore, it cannot be dissolved or transported in the blood. Instead, albumin acts as its carrier protein. Then the bilirubin reaches the liver cells. Inside liver cells, the bilirubin, which is previously in unconjugated form, is conjugated with two molecules of glucuronic acid. Actually, the unconjugated bilirubin cannot be dissolved in blood. Therefore, for its easy transportation, the bilirubin is first combined with two molecules of glucuronic acids. The two molecules of UDP glucuronic acids act as the substrate. It provides glucuronic acid to the bilirubin, forming bilirubin diglucuronide. This bilirubin diglucuronide is the conjugated form. So this reaction over here is really important as it shows the conjugation of bilirubin inside the liver. The conjugated bilirubin in the form of bilirubin diglucuronide is then secreted inside the bile. Then from the bile, this conjugated bilirubin goes into the small intestine. Inside the intestine, the conjugated bilirubin is again converted into bilirubin, and with the help of bacterial enzymes, it is converted into urobilinogen. The urobilinogen is absorbed by the blood capillaries, from where it re-enters the liver, and from the liver it comes back through the bile inside small intestine. This urobilinogen inside the small intestine is converted into stercobilinogen with the help of bacterial enzymes, which is further oxidized into stercobilin. The characteristic brown color of feces is due to the presence of stercobilin. However, some of the urobilinogen does not go into the liver cell and is not returned into the small intestine. However, it enters the systemic circulation, and this urobilinogen is removed by the kidneys. Kidneys filter it out in the form of its oxidized form, urobilin, in the urine. The characteristic yellow color of urine is due to the presence of urobilin. So now we have studied the complete cycle of heme degradation. Heme is converted into bilirubin by the macrophages. Then the liver conjugates the bilirubin with two molecules of glucuronic acid. The conjugated bilirubin enters into the small intestine, where some of it is absorbed in the form of urobilinogen, and the rest is converted into stercobilin, giving the brown color of the feces. The absorbed urobilinogen is mostly re-returned into the intestine. However, a small portion of it is filtered by the kidneys, and the yellow color of urine is due to urobilin. So let's study about jaundice. Jaundice is a condition in which there is yellow discoloration of person's eyes, skin, and nails. The color of stools, urine, and feces also changes. So you can see here the eyeball is yellow instead of being white, and the skin is all yellow. The nails also become yellow. It is due to the accumulation of bilirubin. As we know that the oxidized form of biliverdin, the bilirubin is orange-yellow in color. Either the bilirubin is not being excreted from the body, or it's not being conjugated by the liver, or due to excessive hemolysis, the bilirubin levels in blood increases. So there are three types of jaundice actually. The hemolytic jaundice is the first one we're going to discuss. As the name suggests, the hemolytic jaundice will result due to excessive hemolysis of red blood cells. The liver has a capacity to convert more than 3000 milligrams per deciliter of bilirubin into its conjugated form, which can be excreted from the body. However, If there is more hemolysis due to some disease such as sickle cell anemia or the deficiency of some enzyme such as pyruvate kinase, then there is more hemolysis. More hemolysis will produce more unconjugated bilirubin. Liver is continuously converting it into conjugated bilirubin, but even though liver converts it into conjugated bilirubin, still the levels of unconjugated bilirubin and conjugated bilirubin both will be elevated in the blood due to excessive hemolysis. If there is more conjugated bilirubin, it is increased due to the damage to the bile cells, and the feces will appear normally dark colored. Enzymes urobilinogen will be excessively produced into the higher levels of bilirubin. The elevated levels of ALP in the blood is so the next type is the hepatocellular or hepatic jaundice. As the name suggests, this jaundice would be due to the damage to the liver cells. Damage to the liver cells may result from cirrhosis or hepatitis or any other liver disease. Since liver cells are damaged, therefore, they are unable to convert unconjugated hemoglobin into the conjugated ones. If they can't convert unconjugated hemoglobin into the conjugated bilirubin, the bilirubin cannot be released from the body. In this case, unconjugated bilirubin will be really high in the blood. However, if some of the liver cells are producing conjugated bilirubin, then this conjugated bilirubin, instead of being excreted from the body, would be excreted in blood. So in hepatocellular or hepatic jaundice, unconjugated bilirubin is high in blood. And the conjugated bilirubin is also directly absorbed in blood instead of its secretion from the body. The kidney then removes this conjugated bilirubin. The color of conjugated bilirubin is dark, so the urine will appear dark colored instead of the characteristic yellow colored urine which would be due to urobilinogen. The feces would be clay colored. They would not be normal dark colored. This is due to the lack of stercobilin, as the conjugated bilirubin is not being excreted into the intestine properly. Therefore, stercobilin is not produced and the feces would not be dark colored, instead they would be clay colored. Since there is damage to the liver cells and they are unable to perform their function, some enzymes which are related to the liver cell will become increased in the blood, such as ALT and AST. The ALT and AST, alanine transaminase and aspartate transaminase, both these enzymes would be raised in the blood, and this is a common method used to detect the hepatocellular jaundice. So the last one is the obstructive jaundice or post-hepatic jaundice. As the name indicates, this type of jaundice would be due to the obstruction of bile duct, which can be caused due to a tumor or a stone or any other infectious disease inside the bile duct. The most common symptoms would be the pain in gastrointestinal tract and nausea, along with the yellow discoloration of skin, nails, and eyes. Conjugated bilirubin would be increased in the blood. Since it's not secreted and it's highly soluble, it would be reabsorbed inside the blood capillaries. And if the conjugated bilirubin is not released in intestine, then stercobilin is not formed and the feces would be clay colored instead of their characteristic dark colored. All of the conjugated bilirubin inside the blood is then filtered by the kidneys and it is released in urine, therefore creating dark color of the urine. Another important point to remember about the obstructive jaundice is the enzyme ALP is increased in obstructive jaundice. It is increased due to the damage to the bile cells. These cells secrete this enzyme into the blood. The elevated levels of ALP in the blood shows that the patient has obstructive jaundice. So that's all for today. We'll learn about the 3 types of jaundice. We'll learn about heme degradation.